So it's my pleasure to introduce my colleague Professor Jayesh Bellare on this occasion, and Professor Bellare happens to be the alumnus from this institute. I think he graduated 1982. So he graduated from this institute 1982 from our department of Chemical Engineering, and after graduation he went to University of Minnesota, Twin Cities, Minnesota and Minneapolis. At university, he has been consistently rated as in the top 3 for chemical engineering departments in the US and by extension across the world, I would think. So after PhD from Minnesota, he went to MIT for a postdoc for a couple of years, and later joined our institute 1990, is that right? So 1990 he joined our institute, and from there he's been a faculty in our department. And Professor Jayesh research areas have been at the interface of nanotechnology, microtechnology, biosciences and bioengineering, and he's conducted research extensively, published more than 225 papers, actually more than 230 papers in peer-reviewed journals of repute. Has graduated, obviously has graduated several PhD students, MTech students, and advised many undergraduate students in the department. And he's got several awards. Because today I think we are going to listen to him because he's been conferred with his H.H. Mathur Award, but prior to this he was won several awards, and most notable among them are 3D Graphy Medical Awards 2020. He's got Lifetime Achievement Award from Government of India, Ministry of Ayush, was elected as a Fellow of Maharashtra Academy of Sciences, elected as a Fellow of Indian National Academy of Engineering, National Academy of Sciences India, and several others. And so he's been consistently winning awards. There is a big list, and he's been an excellent researcher. So I think with this brief introduction, I would request Professor Bellare to come and give his talk.
Thank you, Dr. Madhavinjamur, for that introduction. And of course, I should thank IIT and the deans, current and former, and associate deans for the effort they put into these awards. It's been my honor and pleasure to have received this the other day. And what I'll be talking to you about is not a technical lecture per se. It's more of a little overview. It's a bird's eye view of what it's hard to summarize decades of work into. But actually, it's easy when you skim over the top and look at it, and that's what I'm planning to do here. So what we've been looking at over the years is nanostructured materials, and it evolved sort of into healthcare. I'll have 1 or 2 slides to tell you how we got there. So first, of course, I would like to thank my group members. There have been several. Not everybody is captured in these slides, but they are there. I can see some future group members also hiding in there. So hopefully this inspires nobody young enough here to do that, but hopefully it will. So, and I have been very fortunate in excellent colleagues with whom I've had good interactions within the department, at IIT and elsewhere as well. And I'll mention some of those as we go along. So my thanks are to them. As so well-known secret amongst most professors that we are the conduit. All the research work of our students gets communicated. Therefore, it's important to make sure that they get acknowledged, and I here do so with great gratitude along with the funding agencies that helped me. Okay, so this talk is largely about healthcare. So, like other professions, there is a toolkit that a doctor has, and that doctor or the medical profession has 2 pieces in his or her toolkit: drugs and devices. Right? They are traditionally separate. drugs are what the medicine people use, and devices are largely what surgeons use. But those lines are getting blurred, right? So many of these are getting now interchanged, and that's where nanostructured materials have come in here. So for example, take example of stent. It began as a purely mechanical device, still is in many applications, but not good enough today because restenosis happens, and therefore you need to keep it open. These stents have to be drug eluting, and therefore the technology has moved from a mechanical device to what is the best drug to be incorporated, what is the best way to release these drugs here. So what began as a profession or an industry, which was a mechanical industry largely, now is looking at drug delivery. So this is one direction in which it is going. The other direction is from the drug side. Right? The life of a diabetic is difficult, right? Many testings, many drugs to be taken, anticipating what you will eat before you take a shot. So insulin today has reached that level of biotechnology achievement, which is tremendous. It's a microbe that produces it almost entirely, except for a little bit at the end. Epitome of biotechnological advancement, exact duplicate of human insulin. So this is drugs. So it began chemical, drug industry was chemical, became biochemical, but that's not enough. So now today's the drug industry is moving towards devices. So device has to be what the insulin provider has to provide. Began with a pen fill syringe, which dose. Today it is a closed loop control. In fact, closed loop is still a little harder. It's at least open loop control, wherein there is a direct dosing. So there's an insulin pump which provides all this technology, right? So this is where where this industry is going. There is a mixture now of devices and drugs together, and it's not the separate two that used to be. So that's where nanostructured materials help. And here are some examples of effects of nano that are important in the biological realm. Experts in physics and chemistry who work with nanostructured materials look always at a level that is finer than this, quantum effects in particular. But there are biological effects which do not always arise from the quantum effects. They arise from the fact that the human or the animal or biology has evolved largely without exposure to this category of materials, or this size scale of material. I mean, that's a very simplified way of putting that. Therefore, nanostructured material ability to withstand the immune system. So there's a macrophage evasion mechanism that these have, which is very useful from a drug delivery viewpoint. You can deliver things that otherwise you could not as a medicine. These allow us to even cross the blood-brain barrier, which is a very natural barrier that has evolved, present in many systems. And this blood-brain barrier is a barrier that is preventing today's drugs from reaching the brain, except for a few mind-modifying drugs which are being looked at very carefully by pharmaceutical researchers to see how they can incorporate drugs. But turns out that if you make them into nanoparticles, there is a good way to cross the blood-brain barrier for some drugs. These nanostructured materials can also slip through cell junctions, and therefore they can reach tissues where otherwise they would not. Next, the nanostructured materials, especially nanotextured materials, can easily integrate with cells, join to cells, and therefore there is this important field where implants today are nanostructured or coated with nanomaterials so that they would integrate well with the surrounding tissue. Fifth point, many of these nanomaterials or nanoparticles in particular can deliver large quantities because each nanoparticle contains many million, billion drug molecules, and therefore diffusing one particle in there can deliver a large amount. And lastly, point number 6 is that particularly for cancer, for tumor and anti-cancer therapy, there is this characteristic of tumors which is very well exploited by nanomaterials for cancer drug therapy, and that is tumors are very greedy and they take up not only oxygen, they build new blood vessels, they take up anything that surrounds them, including nanoparticles, and that is this phenomena of enhanced permeation and retention. So these are all properties at the biological level of these nanostructured materials or these nanomaterials, and that's where modern nanomedicine is headed today. So one slide of historical evolution. The way we fell into this, or I fell into this, was because of an old interest, right? And I need to tell this because it's an interest which matured as a student at IIT Bombay, right? And that is photography. Interest in photography led me to study electron microscopy later in life, and which is something I do even today. And when I approach the dean through RIFC and other similar schemes, IOE schemes, at the back of my head, I know I'm asking for a fancy camera, right? So that's what it is, except that it happens to cost 16 crores or whatever the latest figure is. But, you know, I mean, that's technical hobby for you, right? It gets imprinted in your brain as a kid and never leaves you. So electron microscopy, especially of liquid systems, is one of the key things that we do. And that has led to many of the work that we do, led to soft nanomaterials, because we could see these soft nanomaterials in an electron microscope to determine structure. And biological materials happen to be soft nanomaterials, really. So interaction of synthesized materials with soft nanomaterials was one of the areas that grew in my group once I came back and joined. The third point is important, and that is, you know, reaching far out. And it was a time when biomedical engineering was, you know, sort of hotting up on campus. It was there for a long time, but there were some events which happened around that time that got medical professionals onto campus, right? For example, even today, we take in medical doctors and give them engineering degree, and we take engineers and scientists and give them a biomedical engineering degree. So that led to many medical professionals come onto campus to talk, to give us lectures, to give our student lectures. And that led to casual conversation, corridor conversation, which led to some of the projects which I'll mention to you about. So this curious combination of situation has really led to our research in medicine and medicinal systems as well, which I will not talk about today. And that is the work that we do with both traditional and alternative systems of medicine, which has been quite path-breaking, I would say. But it has led to a general appreciation amongst the medical profession that what we do could be useful to them. And that is where this applied nature of this work comes about. So we do many things here, and I'll just fly through this slide to give you an overview of the kind of work that we do here. So for example, and some of these I will elaborate in slides to come depending on time available. So for example, we make micro devices for cardiac use. These devices are opposite of the stent example I gave you earlier. Stent keeps a vessel open. Occasionally we want to close a vessel. And these vessels are things could be like, loosely speaking, holes in hearts, congenital defects, blood vessels malformed which need to be closed. Closing a vessel is much harder than opening a vessel, especially from the inside. So that's one area we've been working on. We've been working a lot on bone grafts, and I will talk more about this later. Drug delivery with nanoparticles through the eye, for the eye as well as for other parts. Quantum dots, this is where a little bit of traditional quantum nanomaterials comes in for diagnostic purposes. Nanoparticles that arise in traditional and alternative systems of medicine, which I will not talk about, not today at least. I have lots to talk on that, but some other day. Hollow fibers, which have got very special structure for superior kidney dialysis, artificial organs, based on stem cell bioreactors. So these are some of the ideas that have reached some level of maturity. These are all at fairly low TRL levels if one wants to look at it from that metric. But there are one or two which have reached high TRL level. There are a couple that have, at least a couple that have transferred to industry. There's one that's at 7 or 8 and so on. But these are all offshoots that look like great ideas. Unfortunately, I cannot claim credit for any of these ideas. These ideas came about really from the medical professionals we interacted with. Right? So out of the hundreds of problems that were thrown at us by these medical professionals, these are a few that we happen to have some success with. And there are of course others were not on this slide. So with respect to health, especially with respect to health and welfare, nano has many challenges. The main one is the public perception that whether it is safe. Right? So some of these are issues that have even come to us as parliamentary questions. Is the work you're doing really safe? Are you doing it in a safe manner? And we've had to answer that as well. Then there are uncertain regulatory systems, right? And this is where there is a great barrier for medicinal development, surgical development, because of the regulatory systems here. So the uncertain regulatory systems, they don't know what is a nanomaterial. The regulators have some difficulty there. We try to help. Difficulty in getting cell lines and animal models to do the necessary regulatory, preclinical work. Large animal testing. That's really a hard thing to do, and even for the vaccine development over the last couple of years, this was one of the rate limiting steps. And lastly, the regulators have really onerous requirements, and meeting those. And I'm happy to say that we actually went through this process for one through the IIT system, which is what I'm really proud of. So this is something that is that one needs to keep into mind, that these are some of the challenges that one has to overcome when one looks at applications associated with healthcare plus nano. So I'll give you 3-4 examples of these here. So one of the problems that we were working on, and this was late Rinty's PhD project actually with me, and that was delivering a drug to the posterior chamber of the eye, especially to the retina. Right? Just like a blood-brain barrier, there is a retina-brain barrier. There's a retina-blood barrier, very analogous to blood-brain barrier, and therefore it's very difficult to reach anything to the posterior chamber of the eye. But turns out that if you package the drug into nanoparticles and instill them very simply as eye drops into the eye, there are transport mechanisms which take it back through cell junctions. So you can put it as eye drop, or without invasively going into the posterior chamber, you can inject it into the musculature that surrounds the eye, and you get great results. And we began this as an experiment for age-related macular degeneration, but that's where the medical professionals came to us and said, "Hey, you're looking at the wrong disease here. Look at cancer of the retina, retinoblastoma." Right? And that's what we did. So we made these nanoparticles and we found what their release characteristic was, and then eventually this actually went on to first in human trials at LV Prasad Eye Institute, where we had a collaborator, and Apollo Hospitals, and one of my PhD students who's now working at industry is worked on this. And here is one of the satisfying successes that you have, that it actually reaches a human, and where it shows that there is a possibility of effectiveness. But that's where it ended. Did it save any lives? Or did it save any eyeballs? Because the unfortunate treatment for this condition is removal of the affected eyeball. And you can't inject a drug into the eyeball because the injection tract causes severe metastasis of that cancer. So this is unfortunately, this happens to be one of those orphan diseases. So despite our efforts to interest pharmaceutical industry in this, we were unsuccessful. We had reached a rather high TRL level. So that's one of the frustrations of doing this sort of. It's satisfying. So the joys and frustrations are reflected in this particular project. Yes, it worked. We got good delivery of carboplatin anti-cancer drug right at the retina without injecting it by injecting it only into the surrounding musculature. But what can you do with that? Okay, next example. So this is an example of delivery of a drug to the lung, and this is a curious example. It's curious because all of our lungs undergo a massive change in surface area, and as any elementary physical chemistry calculation would do, there's a surface tension associated with that, and therefore there's a lot of work to be done as the area increases. And this work is what is part of the basal metabolic rate that we all have. And this work is reduced in all of us by having a surfactant, right? Essentially a soap which coats the lung, and this has now been implicated in the pandemic as well, as being one of the causes for this for the lung getting affected. So the basic problem is, how do you ensure that the work of breathing is low? It is for all of us, except in one case. And that case is exemplified by this cartoon here. It's a nice cartoon which hits the problem head on. What's this cartoon? This cartoon shows a child just born, and the obstetrician who has delivered this child is about to smack it on its bottom. And they do that to get babies breathing if they don't. And in fact, the lusty cry of a baby contributes to the APGAR score, which is a way of rating how healthy a newborn is. And of course, the cartoon part of this is the fact that there was, I think this came out at the time when there was corporal punishment law, which came out, and he's saying cops. Right? So that's the joke. But the fact is that there are premature babies born where this surfactant, which we all have, is not yet developed, and therefore causes a severe breathing problem. And we need to do something to tide over a few weeks until the immaturity gets over and the lung surfactant builds up. It's a natural product of the growth of the baby. So solution is simple. You make a surfactant nanoparticle, formulate and with certain adjuvants which activate the breathing centers and so on, and you can just inject it as a bolus into the newborn or the baby's nasal cavity. It goes in, spreads out, reduces surface tension, and solves that problem largely. Allows the baby to tide over a few weeks until her own surfactant starts building up. And using cryo-electron microscopy techniques, we were able to show the structure of these particles and how these particles break upon hitting walls. Of course, this is in the microscope, but that's an analog to what would happen in the real world. So again, this was something that we developed, showed in the lab, and it's been a struggle to take it since. Yeah. Right? Professor Denti tried for a long time, and I think to a last day she was still trying. There were some successes to bring it into some animal studies. The problem here is that you need premature. I mean, imagine doing an animal trial on premature pigs. Which animal ethics committee is going to give you permission for that? Right? First of all, it's a large animal. This problem with larger animals, you can't do small animal study. So again, this is in some ways ended in frustration. What do we do with a project like this? But that's where it is. So, okay. You don't give up, I guess. We'll still try. The next thing I'll talk about is these little tubes. These are hollow fibers. These are tiny fibers. They're small tubes, but they have a very complex structure in the wall. The cross section of these tubes have got a filter built into them, and the filter has a graded porosity going from large pores to small pores and so on. So these are hollow fibers, and we can make these are useful in many things here. We got into this because of a demand for kidney dialysis. Right? This is the core technology associated with a dialyzer, which is used in kidney dialysis, and that spun off various things here. So here the nano angle comes from the fact that we are able to tune the structure of the polymer by using additives into these little nodules, and the gaps between the nodules enables efficient filtration. The additive that we put coats sticks out of the nodules and provides a very biocompatible surface. And some of these we can image with high resolution SEM to show that this nodule size is appropriate. And it also grows cells very well. Biological cells can grow on the surface of this hollow fiber membrane or hollow fibers very well, allowing this now to become a biocompatible surface on which cells can grow. And then you can do all sorts of fun things with it. For example, you can, without growing any cells, you can build a regular dialyzer. By growing cells, you can try and make an artificial kidney. And the artificialness of the bioartificialness of this comes about by virtue of the fact that many of the middle molecules, so to speak, which need to be reabsorbed, which is traditionally done in a natural kidney, are not done in dialysis, but by putting cells on it, we are able to do that. You can convert it into, by growing cells of a different type, pancreatic cells or stem cells modified into pancreatic cell line. You can make it into a bioartificial pancreas, and this has gone on to animal trials where we showed that in an induced diabetes model, the normoglycemia is restored, normal blood sugar is restored for a period of 30 days by implanting these into the peritoneum. And this can be implanted with a standard injection into the peritoneal cavity. And this there's industry interest in this, and we are trying to see how this. So there's a JDA with a company here in which we are trying to see how this can be taken taken forward. So idea is these hollow fibers have many such uses. Another one is the bioartificial liver, which has been tested in the lab, but now is waiting, trying to see whether we can do some animal experiments, which are always hard to do. Whether or not you have an animal house, which I hope we will soon have. The next example I'll give you, and I think I have one more after this, is this problem about poor plumbing in the heart. Poor plumbing in the heart arises usually due to developmental difficulties in utero. There are several. One of them is one in which there's a small duct called the patent ductus, which while the baby is in utero, short circuits flow from the lung because the lung is not functional. I mean, it serves no function because the oxygenation happens through maternal blood. But the moment the baby is born, this duct has to close. So the short circuit has to close. And that's one of the many events that happens within seconds or minutes of birth. In a few cases, it doesn't close. Right? So that's one type of defect. If it doesn't close, then the blood bypasses, short circuits the lung, poor oxygenation. And there are many other such defects. For example, this wall sometimes has a hole in it, and there are other defects of this sort. So this is a problem that we try to solve, and the solution is through nanobio and microengineering. And the idea is to use a catheter, just like one would do for delivering a stent, except that now you deliver something which will open out and close that opening you want to close. So the design which we came up with was a double umbrella sort of design, and we had a nanomaterial that was coating and filling this umbrella. So the stoppage or the occlusion was caused not by this material itself, because it's a rather porous material, but by the fact that it promotes local clotting of blood, and that thrombus is what actually stops the flow. So this is technology which causes this clotting cascade to be initiated, and this clot is what actually prevents, very natural clot which prevents, and that clot of course should not spread out too much, and that's the trick there. So we developed this, we patented this, this was done in collaboration with a cardiologist, interventional cardiologist who's an expert in this area, and he was very happy with the performance, until we thought, we'd be able to interest industry in it, or we may even, this was now the era where startups, were growing, right? Startup was a bad word when I joined, but I think now it has gone to the other side. I don't know what's the opposite of a bad word, but whatever, it's that word, now. But I'll come to that in the next application. So this is again another one of those frustrations wherein you do, the joys you prove it in the lab, but the frustration is what do you do with it, right? Thereafter. So that's a gap that we need to fill somehow. So we proved this, and the cardiologist was happy with it. We proved it in the lab, of course. Imagine now doing this experiment for regulatory purposes in an animal. How can I get a pig with holes in his heart? A baby pig at that. Okay, enough said on that. Okay, last example I want to give you one is one on bone. So sorry for this gory photograph, but this was shared by Dr. Vivek Soni, who's a maxillofacial surgeon, and many such bone loss cases arise. And they arise simply because of trauma. I mean, that's the most common. This is an extreme example, but fractures are very common, all sorts of complex fractures. Turns out that even extraction of a tooth results in bone loss surrounding the extracted tooth, which prevents an implant from being put in. So bone is something that is that's an important organ or tissue of the body. And in bone tissue engineering, what we are seeking is a way to replace part of a bone that's taken out, and that part has to be replaced with a material which has this complex morphology. And we want this to be something that is not a metal. In all these examples I've given you, we don't use metals or we don't use anything which is a long lasting, because there's a general feeling that anything that lasts long in the body could create a problem in the future, and it has been proven so. For example, big multinational had to cough up millions of dollars, and they're still trying to settle it in India when a knee implant had issues with it. So we work with materials that go away with time, and with bone it's great because bone heals itself over a period of time, no matter what the age of the patient is, usually, unless there's some very peculiar diseases, but in most cases bone heals. So we need a temporary bridge, right? It's a temporary bridge over, and that's what this bone scaffold does, and that's what we make. So, and we make this by mixing whatever natural bone has essentially, except that it's human made. Hydroxyapatite, which is a form of calcium phosphate, a ceramic material, gives some strength, and there's a polymer in natural bone, largely collagen. Many people use, many of the scientists in this field do use collagen. We took a different approach. We use gelatin, which is a much cheaper form of, comes from the same basic molecule, slight change in structure. And other colleagues of mine have, and his students have looked at gelatin as a material on which things can grow, and they find that even for other tissue, it's a great material. So basically what we do is that we take gelatin and some other polymers, chitin-based polymers, chemically modify them a little bit, mix them all together, and then process them in a way that gives a nanoporous structure, exactly like how a bone might be. And then we can do all sorts of things. We can even 3D print it like this. So we can take a CT scan and figure out what the defect is, do a virtual surgery on this, and then build one of exactly that shape. In fact, this is a case in which this part of the chin is going to be removed by virtue of cancer in a Tata Memorial Hospital patient. And this is a graft which we have made. Unfortunately, it's not going to be actually used because, again, because of the regulatory issues, which we are now trying to comply with. And then there are many long bone defects and so on. So what we do is to make these materials, and these materials are resorbable. They are biodegradable in the body. They go away after a period of time as the bone grows through. So we leave these hollow pores into which the natural bone grows. As the bone grows, rich source of calcium, what we've put in, and this material eventually goes away. We've shown through various studies, all published work, many of the regulatory things we were able to do, showed it in long bone. We've shown how in a treated sample there is very early healing. So this is, for example, week 14 of an induced fracture done with due ethical permission, which shows very good healing when treated. Without treatment, although the body tries to heal it, it cannot bridge the gap. Right? The river is too wide for a single swimmer to get across here. So you provide this scaffold, you provide the floats, and the swimmer can go across and produce, heal this defect. And this has led to a range of products. A bone graft is one that we are about to, well, we actually have finished the first in human trials after a great deal of effort, including setting up a GMP facility and getting the drug controller approval. And there are many other such things which are in the pipeline. So this is one example where we got it to first in human trials. We did a pilot study, and now we'll be starting a pivotal study, and hopefully there will be a startup nucleated around this soon, which we will take through sign. So I hope I've shown you some of the joys and frustrations in doing this research and translating some of these innovations from bench to bedside. And just to summarize, so all the materials we have used are resorbable materials. They have important applications in many regenerative medicines. There are special facilities needed. Good manufacturing practices is one. It's like electronic grade clean room, except you do biology in it. Point number 3, I did not elaborate on, but we've shown that nanotechnology, nanoparticles are a unifying theme across modern medicines, allopathic medicines, as well as Ayurvedic medicines and homeopathic medicines. And this line of research can perhaps revolutionize some of the healthcare the healthcare delivery issues for many of these societal problems that we face today. Thank you.